thank you all for coming this afternoon. Especially thank you to Dom Delvilla and to Sarah, my mentor. My name is Will Coburn. I come from Allen Hancock College, Santa Maria, and my major is electrical engineering. My mentor for the summer was Sarah Grandin, and our faculty advisor was Dr. Luke Garajan from the Department of Electrical and Computer Engineering. The primary funder is National Institutes of Health, and this summer I did research with micro-contact printing of polyol-lysine using PDMS stamps for the adhesion and patterning of neurons. Now, before I get into that, why would a group of people want to do research on neurons? Well, let me pose to you a big global problem that's facing a large percentage of the world today, and that's neurological disorder or disease. It's such a big problem that the World Health Organization in 2007 did a study, and they found that 1 billion people, about a sixth of the world's population, suffer from some form of neurological disorder. They also found out that Europe spent around $200 million in 2004 alone on palliative care for treatment of people with neurological disease. And they also found out that effective care is largely unavailable for people who are suffering with neurological disease. So we can see that we want to find out as scientists to do research, to investigate how neurons work and how damaged neurons work. And so maybe we can find a cure or an avenue to a better way to treat these disorders. So before I go further to the research, what is a neuron? A neuron is a brain cell, and we have billions of those, around the average about 100 billion in our brain. They're very small. Their primary purpose is to send and receive information with each other via electrochemical signals. They are in our brains, and they regulate who we are as individuals. They regulate how we speak, how we think, how we act, our sensory reception, our motor function, basically who we are. Here's a blown up version of a nerve cell. Here's the cell body, or the soma. Basically like any other eukaryotic cell, undergoes the same metabolic processes. They do have distinct characteristics though that set them apart from other cells. They're called neurites. These are dendrites and axon with an axon terminal. The dendrites receive the electrical stimuli from other neurons. So they receive the information, just as two people would be verbally communicating to one another. They receive electrical stimulation. The soma assimilates that information, and they send out an appropriate action potential or an electrical response, and it's emitted out of the axon terminals to other neurons to get the information. And so it goes moment after moment. Right now, as I speak, this is happening millions upon millions of times. Here is an artistic depiction of a healthy neuron, and here's one of a damaged neuron, say by a motor neuron disease or Alzheimer's. Everything is degenerated, and so when the structure degenerates, the internal degenerates as well. And the internal is the electrical signal. So we want to, in this case, study the electrical potentials from the extracellular neurons, so we can find out discrepancies among healthy and damaged neurons. So the research goals for the summer was to create a novel and reproducible way to stamp neurons in a pattern so we can quantify their electrical behavior on glass substrates and eventually multi-electrode arrays, which is right here. We would stamp these neurons, this is a blown up picture right there, on this glass substrate, and this glass substrate has embedded electrodes to record the electrical potentials of the extracellular fluid around the neurons. Then we wanted to incubate neurons and record the neurite growth. As you can see some neurite growth here, visually. So in order to plate the neurons, we use a polydimethylsiloxane stamp, or PDMS stamp, and it's basically an inert organic substance, much akin to rubber. It's translucent, and we cured it. It was 1 part base, 1 part curing agent, 10 part base. We cured it, and we baked it, and we cut out small sections of the stamp that had 3 to 7 little square-like geometries of these pillars. And the pillars ranged from 30 to 100 microns in pitch, or center to center distance, and they were 15 microns in height. and the diameters ranged as well. And so what we did is we coated, or we inked, this stamp with polyl-lysine. It's a hydrophilic polyelectrolyte that acts as a cell adhesion promoter. So when we would ink this for about 30 minutes, we dried it, and it would leave a residue or a monolayer of this polyl-lysine on the upside of these stamps. We would invert it, then apply a force to get these geometries on the glass substrate of this polyl-lysine. And in theory, these neurons, these cells, would adhere to these specific spots, and we can quantify their behavior. So there were problems. The main problem was pillar deformation of the PDMS stamp. It is an elastomer, so if we apply an excessive force or an inordinate force in some portions, it may experience some bulging or some buckling. Here is an actual representation of what the pillars would look like if they did buckle. These are the color ones outlined. And it would, in consequence, we have good deposition here, but here is lacking, and there's some smudges. So this deformation, this pillar deformation, just is not, it's not a, a reproducible way to do things. So we wanted to find out some solutions, and so we realized that PDMS is an elastomer and it can be deformed pretty easily. So we thought to glass, maybe glass backing. We can't use glass or silicon stamps as it wouldn't deposit PLL properly, but here, a Young's modulus is basically the ratio of stress to strain. It's basically the stiffness of any given solid or material. So we found that the Young's modulus of glass was 5 orders of magnitude higher, and so glass obviously is stiffer than PDMS. So we decided to back and bond the glass to the stamps, the backside of the stamps, not where the pillar geometries are. And we also decided to implement some quantifiable force or a weight on the stamps itself, so we used some weights. So after it's plated, this is an ideal version of what would have been plated. We put the stamp substrates that were stamped in 6 well dishes, and we mixed the neurons with some media, so there's about 2 milliliters of media mixed with 100,000 rat hippocampal neurons. So these neurons are very new, very young. They don't have neurite growth yet, and this is what it would look like. And so what we did is we took an unmodified stamp, or one that wasn't backed with glass, and this is 9 days in vitro, and just with the force of the finger, we found out that there's a large overgrowth here and absence here in some areas. Large overgrowth here, and the pitch between spot to spot was 25 microns here, 35 microns here. So large overgrown areas, polyl-lysine absent areas, and so we just deduced that it's non-uniform distribution of force. So we wanted to see how the weights were a little more uniform. This is 12 days in vitro with a 200 gram weight on the stamp instead of just using our fingers. So we got a little more uniform distribution of polyl-lysine stamping, but there's still some overgrowth, and there's still areas where there are no neuronal growth. So we deduced an indication of pillar deformation. So what we tried was the glass back, and we see here that most of the sites where we had a monolayer of polyl-lysine was deposited on these glass substrates. There's still some overgrowth of neurons, which could be attributed to not completely drying the stamps or over deposition of the monolayer on the stamp for stamping, but pretty good deposition of polyl-lysine. Here's a good one. This is an ideal one that we did. A little overgrowth, but we want it basically to look like this. We have neurons adhering to the PLL, and we have neurite growth, the dendrites and axons, neurite growth between them. So this is an ideal thing. So we found that backing it with glass we found it more reproducible and better results. So obviously this research is in the beginning stages, somewhat relative. But we want to have an ongoing research with plating these neurons on multi-electrode array. So like I explained before, we want to take signals, electrical signals from these neurons, and if we know how normal neurons work from diseased neurons, maybe we can find an avenue to cures for some neurological diseases. We also want to explore using atomic force microscopy to directly deposit PLL onto glass substrates and MEAs, so it could make it more robust and more automated, so that we don't have to go through stamping and all the errors and everything. This would make it automated so we can move on with the research. So that's all I have for today. Thank you very much for listening.